just while people are joining, I was going to say hello and welcome to Imaging One World. And if anyone wants to join the organisers, then please give us a shout, because we're looking for new volunteers to help organise this moving forward. And also we're after ideas of any talks for next year. If anyone has any speakers, then any speaker ideas, then please let us know, because we're trying to plan next year's schedule. So today I have great pleasure in introducing Jan Huisken, who is going to talk to us about, actually I don't know what you're going to talk about, but I'm hoping it's about your light sheet work and where the developments you've done in the light sheet field, which have been quite amazing over quite a number of years. So I think it's probably better if I hand over to you and you start and actually tell more about yourself, because you'll do a better job than me.
So can you see my slide?
Yep, that's perfect.
Thanks, Alex. Thanks for the introduction. Thanks for the invitation. A great pleasure to be here. So I should maybe briefly mention that I'm a physicist by training who runs a multidisciplinary lab here at the University of Göttingen for about a year now. I was before at the University of Wisconsin at the Morgridge Institute for Research, and recently moved to Göttingen. And so most of the results I'm going to show you today are actually results from Madison, and even some from Dresden, where I was before. And now I'm building up a new lab here. And so hopefully I can, towards the end of the talk, I'll share some impressions from what we're doing right now here in the lab as the lab is being renovated and we are filling the space slowly with equipment. So my talk is going to be split into about 3 parts. So the first part is a general introduction into multiscale zebrafish imaging using light sheet microscopy, which also includes a quick introduction into light sheet microscopy for those of you who are not familiar with it. And then in the second part, I'll discuss briefly how I think sustainable microscope development could be done in an optics lab, and how this allows us to build more reproducible instruments in our own lab. And then in the third part, I'll go into the sharing, open science aspect of all this, sort of how we think we can more effectively collaborate with biologists by making our instruments more accessible for users. So on to the first part. So just as a quick introduction to the questions of the lab. So we basically, as I said, a multidisciplinary lab, mostly biologists and physicists. Biologists come up with some interesting, exciting questions, and then the physicists try to build the best possible microscope for these questions. And so on the biology side, it's mostly sort of this multi-scale aspect of development, whether it's organogenesis or embryogenesis as such. So we're asking questions how cells form tissues, how they group themselves into organs, why the organs are built the way they are, and how this always happens in a reproducible fashion. And ultimately also how does one embryo differ from another, which I think is an interesting question that can really only be addressed by systematic quantitative microscopy. So, and then on the microscopy side, as I said, it's mainly the task to build the best possible microscope to gently image a fragile object, which is an embryo in our case. And it's not just about the optics and building the microscope, but it's also a lot about programming the microscopes. And there we're trying to move into more automated microscopy acquisition modes, which we call smart microscopes. The big challenge to also systematically analyze the images. So our favorite organism of choice is the zebrafish. We've done other organisms as well, as you will see, particularly with the flamingo. But just as a way to introduce the system here, this is what an adult fish looks like. And then the embryos have this fantastic feature that they're fairly translucent and can be imaged quite well in a also the fish can produce hundreds of eggs every day. And so that opens up the possibility for some high throughput imaging if you want, as long as you find ways to also process the data in a high throughput way. So, as I said, this is a truly interdisciplinary challenge, and we're basically trying to gather all these different people with their various expertise in my lab, but also a lot is done in collaborations, especially when it comes to some more of the more exotic organisms. But still, the task is to bring all these different people together around one table, so that we can come up with some clever ideas on how to do maybe some novel biology. So for those of you not familiar with light sheet microscopy, very briefly, it's essentially a setup that consists of two distinct optical paths. One is used for illumination, and then perpendicular to that typically is a detection path. The illumination path projects a laser light sheet into the specimen, a thin volume in this fluorescently labeled sample, and then the detection lens captures the fluorescence and images this plane onto a fast and sensitive camera. So in reality, this is what it looks like. There's typically a chamber built around the objective lenses that holds the medium. Most of our specimens are held in aqueous medium. And in this particular case, you also see that there's two illumination lenses, essentially illuminating the specimen from the left side or the right side. Yeah, you can see the light sheet in the middle. This is exactly where you would want to place the specimen. And then by moving it slowly through this sheet of laser light, you can then acquire a three-dimensional data set. On top of that, you can also rotate the specimen and look at it from different sides, which sometimes becomes necessary if you're imaging something that's rather large, where simply the penetration depth is not sufficiently good to penetrate the entire specimen. So if you zoom out a little bit more, of course, there's many more components to this, but you may recognize the two objective lenses. Here's an illumination lens. There's actually another one hidden here. Here's your detection lens. And then you have a detection path that typically consists of a filter wheel or a splitter in this case, and a tube lens and a camera. And then the illumination can be done in various different ways, just illustrating that it is a fairly compact, but still in most cases it's built on a large and heavy optical table. So if we jump ahead, and I'll show you just a teaser, a data set that one can acquire routinely, like in this case, these in height. And this is just a simple maximum intensity projection of a time-lapse movie that runs for about 15 to 20 hours typically. And what's striking here is not necessarily only the spatial resolution and temporal resolution, but it has a lot to do also with how the specimen is prepared. And so in order for this experiment to be successful, into creating an environment for this fish to grow happily inside the microscope, and then so that it doesn't move during the acquisition and it still has enough space to actually grow and develop. And so that's always a tricky trade-off to be made. So if you ask me to summarize sort of what the key features of light sheet microscopy are, I'd say it's probably these points. So it's definitely the very low phototoxicity because you're illuminating only one thin slice in the specimen. Secondly, it's the fast image acquisition. You can essentially use any camera you want. And so there's plenty of fast cameras available to capture the fluorescence. The fact that we are so effective with how we excite the fluorescence means that you could even run the camera at thousands of frames per second if you wanted to, because you're not doing much harm by increasing the laser power, because you're only resting in one place in the specimen for a very, very short period of time. And then third and fourth point are really more about sort of the, how easy it is to build and maintain these instruments. So they're very affordable, I would say, compared to a lot of the, especially commercial systems, and also many of the more advanced sort of high resolution systems. And they are fairly robust, as you will see. We can even ship these microscopes around if they're built properly. And then the fourth point is that we think that one strength of light sheet microscopy is that it can be built around the specimen. And that's very important, because light sheet microscopy works along many scales. It works for very small specimens and very large specimens, up to centimeter size clear tissues. And so there's no single instrument that can image all of these different specimens. So as you've seen, most of what we've done has been really sort of this whole organism level imaging, which may be sort of below a millimeter up to a few millimeters. And that's where this Gaussian light sheet, the classic light sheet microscopy, the way we've done it is quite successful. On the other hand, also in clear tissues, light sheet microscopy is the tool of choice. It's basically the task of imaging a centimeter-sized object quickly. And if you used a confocal or so, it would take forever. With light sheet, you can do it in a reasonable amount of time. And of course, things like phototoxicity or photobleaching still play a role. And of course, the ability to rotate the specimen or illuminate it from different sides, etc., may be very important there as well. But at the same time, light sheet microscopy has also been used for like individual cells or small cell aggregates, where you sort of below the 100 micron limit. This is mostly termed lattice light sheet, or at least that's where lattice light sheet has had the most impact. But in fact, you can do most of this also, I would say, with a classical Gaussian light sheet. But this is just to illustrate sort of the wide range that light sheet microscopy can cover, and that there are indeed many, many different implementations of essentially the same concept, just different ways of shaping the beam, or different chambers, different lenses, etc. So in my lab, the multi-scale aspect, basically, if you look at the zebrafish, ranges from like the first few cell divisions, as is shown here, where you can simply look at the embryo from the top. You're looking at the animal pole, you're watching the cells divide. In this case, we were interested in spindle scaling. And you do this, for a limited period of time until, you've seen enough of this of these cell divisions happen. If you then go a bit later, where the embryo is more elaborate, is more complex, and is actually, much bigger also. So in this case, we have around 1 millimeter in diameter here. And again, a time-lapse maximum intensity projection. The color code here is just for depth. But you can see there's a lot going on. And this is a data set that you can only acquire if you're actually imaging the embryo from different sides. Double-sided illumination, double-sided detection, and also rotating the specimen throughout the time-lapse. And this is something we've spent a lot of time on, trying to optimize these kind of movies. And still, I think the challenge here is to be able to image something not just quickly, but also compress the data quickly, being able to image not just 1 or 2 embryos, but ideally many of these embryos, to be able to come up with some sort of a systematic imaging scheme, which also leads then to some sort of a model, a blueprint of how the embryo develops. If we make a big jump ahead, at some point, as the fish grows into a larvae and becomes larger and larger, at some point we simply don't have the tools anymore to do any live imaging. The fish is not transparent anymore, and then we have to turn to fixing and clearing the specimen. And of course, the task in the future is to push the boundary further and further and image longer and longer, and then ultimately use the fixation and the clearing just as the last resort, sort of maybe the last time point of your time lapse, and then ideally, of course, combine the information of the fixed and cleared state at the end of your time lapse with the temporal information that you've gathered through a time lapse. So another thing I'd like to highlight in this context of multi-scale imaging is that you might, in your experiment, you might only really be interested in a certain aspect at a given time. May it be like blood flow in these vessels, but you have to be aware that all of what you're doing there is always in the context of a living organism, right? A living, growing, developing organism with a beating heart and blood flow all across the embryo. And there's a lot of things going on that you simply cannot necessarily capture all at once. And that's something we have to accept somehow, that we have a limited pixel rate. We have a limited field of view that we can capture at a given time. And we simply cannot be everywhere at the same time, right? So something like this entire larvae that's about 3 or 4 millimeters long, you need to acquire multiple fields of view and then stitch the data. And so that means it can only be done in sequence, right? You cannot be at the same time everywhere. And in fact, to get more or less isotropic resolution, you anyway have to image it from different sides, and that can only be done by rotating the specimen. So that also takes some time. So that means we need to be more clever about how we acquire the data, right? We cannot simply blindly, go somewhere and image something, because we might be missing other things that might be going on outside of our field of view. And so, for example, if you were doing a time lapse, then obviously, in the beginning of the time lapse, your embryo may be rather small and compact, but then as time goes by, the embryo will extend and grow. And so in order to capture all this, your microscope has to do some tracking of the object and dynamically increase the field of view and increase the number of tiles that it's acquiring. That's obvious, I think, but at the same time, you may realize that not every tile contains necessarily important information. And so it becomes a challenge then to make a decision what to acquire, when and where. And that's sort of this question that's driving us already now for a number of years. How can we optimize the image acquisition routine in such a way that we have less overhead and more information per megabyte or terabyte, if you want? And so this is a main question of ours that's gonna be with us, for the coming years, I'm sure, or decades even, because it's not trivial to find the optimal image acquisition parameters for all these different questions that we're trying to address with these microscopes. So I just want to give you 2 quick examples sort of where this customization of the microscope plays a crucial role. As I said, we like to say we build the microscope around the specimen, and these 2 examples I think quite nicely illustrate this. So one is the high-speed imaging of the beating heart. So the zebrafish heart consists of these 2 chambers that you see here. There's an atrium down here. The endocardium is in green, the myocardium is in red, and then there's a ventricle up here. And there is actually a way to image this quite nicely at high speed, but typically these movies are 2-dimensional, of course, right? So you you essentially place your light sheet in one plane inside the heart. And so you would get a cross section like it's shown here on the right. And then you simply let your camera run at whatever speed it provides. In this case, it also has a splitter that splits the red and green on the same chip. And so with that data, you can ultimately actually reconstruct the heart in 3D. And there's a trick you can do, which is basically exploiting the feature that the heart is doing a very reproducible movement that can be very well synchronized with the acquisition or the heartbeat in the actual recording. So you can either trigger the camera at a given time point every time the heart contracts, or you simply blindly take the data and then in the end, you extract one heartbeat per plane and then synchronize the data and reconstruct it in 3D. So your raw data simply looks like what's shown here on the right. So one cross-section through the heart at a given depth in 2 colors, 400 frames per second, and you would typically record something like 2 or 3 heartbeats, right? And then if you do this all throughout the heart and synchronize these movies afterwards, you can achieve such a 3D reconstruction that's shown here on the left. Okay. So we've done this extensively by imaging hearts at an early stage, where in the atrium we can very nicely observe this interesting phenomenon that when the myocardium contracts, it folds the endocardium in an interesting, very reproducible pattern, namely in these folds that are held by these tethers that we can find throughout the atrium. And so with this technique, we've been able to get what we believe like a really nice high resolution, high speed insight into the mechanics of how the heart contracts. So again, this I think is a beautiful example that you have to be able to design the experiment around the question. So I don't think you can easily acquire such data with a commercial system. You would probably need to have access to the software to, for example, acquire these movie stacks. So acquiring movies at a given step size throughout the heart. And then, of course, you need software to also take all these data and combine them into this 3D reconstruction. Another example on a slightly larger scale is this gastrulation, which I already showed you briefly. But here I just want to emphasize the point that if you're trying to image an object that's a millimeter in size, you can essentially assume that you will never get a perfect view by just looking at the object from one side and illuminating it from one side. So instead, what most people do in the light community is they do what's called multi-view imaging, where you're not just capturing a data set from the front, which is shown here on the left, but also from the back. So in this case, we have 2 detection lenses. So as you're sliding the specimen through the light sheet, as shown here, you realize that the back camera may capture a nice image of the back and the front camera of the front. And then also if you're able to illuminate from one side and the other side, you can also capture very well, here the top part of the embryo, and in this case down here, the bottom part of the embryo. So essentially, it takes all these 4 data sets, at least these 4, more is always better, but at least these 4 data sets to arrive at a nice reconstruction of the object. Now, the point, though, here is not so much the combination of these data sets, because you have to imagine you're acquiring 4 data sets, you're then merging it, so you end up with essentially 5 times the amount of data that you typically would need for just one object. But it's more about the ability to repeat this experiment more often. So if you do this throughout zebrafish gastrulation, you easily end up with like 10, 15 terabytes of data. And we thought that's not the most efficient way of doing it. It would actually be more sensible if you know exactly where the good data is, where the high resolution information is on your embryo to only collect the data that you think is going to end up in the final reconstruction. But not even that, we decided to reject all the other data and do the merge basically in real time. So if the sphere is, if the embryo can be considered a sphere, you pretty much know exactly which parts will be nicely imaged, and you can place these directly into a map, which is kind of like a world map of the embryo. And if you're doing this for all the individual views, you end up with a reconstruction of the object that gives you a complete picture, essentially taking all the high resolution parts of the individual views, right? And so that means that your time lapse that typically would be terabytes of multidimensional data is now actually just a few hundred kilobytes of a little movie that very nicely summarizes everything that's going on in this embryo, right? And so, of course, there's a number of assumptions made, but critically, this is a way to compress the data so efficiently that also visualizing the data becomes a lot easier, or further analysis becomes a lot easier. But most importantly, you can go on and image the next embryo right after this experiment, right? So you can do more high throughput imaging, if you will. That is ultimately needed if we wanted to compare these cellular patterns in different embryos. So what I didn't mention here was that this is only the endoderm. It's only a single cell layer in these embryos. If you were to image all 3 germ layers at the same time in 3 different colors, then you essentially have to give up this concept of imaging these individual layers and do the spherical projections. You actually have to do the tracking in 3D. But very quickly you realize that the tracking, the segmentation and tracking is pretty hopeless because the data is never going to be perfectly high resolution, and the tracking is never going to be perfectly accurate. And so what you end up with is a big data set of all sorts of quality of tracks. Some will be shorter, some will be longer. And so one way of visualizing this data was this idea to cluster the tracks, these tracklets, and that way get a more complete picture of where the cells are actually moving, right? And so again, it's a way to compress the data and visualize it at the same time, with the ultimate goal to do more throughput imaging, right? Because now you're not faced with large multidimensional data sets, but instead, you have something that's easier to digest and to plan the next experiment after this one. So again, it's all about optimizing the microscope for a given task, and these microscopes may look very different, even though they may conceptually use the same principles, like maybe a light sheet microscope. So that brings me to the second part, which is the sustainability of how we develop microscopes. And when I moved from Dresden to Madison, we realized that we've gone off in all sorts of different directions, but still using light sheet microscopy as the main tool of choice. But still, every little experiment was really an experiment on its own, right? And that meant that all of these setups, each and every publication, was essentially done on a new device that was built by a PhD student or a postdoc, and it was by no means reproducible, right? Each setup would be run by a custom software, and there would really be no benefit from one system sort of carried over to the next system if somebody had implemented some new software feature. So each system was its own system, and it would be incredibly difficult to actually disassemble these systems and move them anywhere, right? So, for example, we came up with this idea to magnetically manipulate specimens, where we injected magnetic beads into the yolk of the fish to rotate specimens in the microscope. And now this is what it looks like under a stereoscope, but then in the light sheet microscope, you have these electromagnets arranged around the specimen, And by turning them on and off, you can rotate the specimen. Now, this is essentially a standard light sheet microscope. It just had this additional feature of these electromagnets. But the way we did it, it was essentially a whole separate setup on its own, right? And the same with optical projection tomography. We also combined light sheet microscopy with optical projection tomography, something you can in principle do on any light sheet microscope. All you have to do is rotate the specimen, slide it through the focal plane, collect these transmission data, and then you can reconstruct the object very nicely, which is this gray contrast here in addition to the yellow fluorescence from the light sheet. So again, this was a separate setup in the lab, but if this had been done in a more, more modular framework, so to speak, then this could have been implemented on any light sheet microscope and wouldn't necessarily need to be done on a dedicated separate setup. So when I moved to Madison, the idea was then to design a reproducible microscope framework, something that would be easy to reproduce, something where you could say, okay, now that I've built one of these, it's easy for me to build 6 more, right? And of course, that's fantastic if you want to do sort of the similar kind of experiment on all these different setups. However, if you can change the configuration of the microscope at the same time, that would, of course, open up the door to do different experiments, maybe even with different organisms, still using the same framework, the same, modular components of such a microscope. And that's exactly what we did. So with Rory, Todd, and Joe, a fantastic team of engineers, I was able to have this Flamingo idea come to life. So Flamingo is just a crazy name for a cool sort of modular microscope concept that allowed us to build microscopes in different geometries. And key to that was, of course, also some interesting optics. And very briefly, we figured out ways to do the multi-sided illumination in a very, very compact form, which also can be done, which can also be used to do the pivoting of the light sheet, the multi-directional light sheet to eliminate striping. And so there's some really neat optics inside the instrument, but I think the key feature still is mostly, the modularity of the system, as is shown here. So you have your specimen in the center, you have a chamber around the specimen, and then you have an illumination arm here on the left, and you have a detection arm on your right, and then you have a set of stages on the top that hold the specimen. And so this ends up being a very, very compact system that is not much bigger than a stereoscope here. And as I said, it's really built around the specimen. So if you zoom out from your zebrafish in a tube, you can see, in this case, it's 2 illumination arms and 1 detection arm. You can see how these individual modules are simply arranged around the specimen. And so what that means is that if your sample rather likes to be in a dish, you can still use the same components, building upwards from this dish around the specimen, in this case, a single illumination and a single detection arm. So there's only a handful of parts that are different between these two setups, but the core components, the key elements, are still exactly the same. So these actually exist. So here's a photograph. On the left, you see what we call a T-SPIM. So the double-sided illumination and a single-sided detection in a horizontal fashion. And then on the right is what we call the V-SPIM, which is ideal for imaging something in a dish. And so you can now play the game of changing geometries and numbers of lenses, and ultimately you can see there's a great variety of different systems that you can very easily build from these individual modules. And that's exactly what we did. And as you can see here in my lab in Madison, we had a number of these flamingos sitting on the table side by side. And The reason here was primarily to have a selection of different systems available to whatever the collaborators would need or we would need in the lab. We could simply pick and choose whichever configuration is best for a given task. And even better so, these systems sometimes were simple skeletons. They would just be sort of the geometrical configuration, but the actual sort of expensive parts, the camera and the stages and the lasers, would only be added to the system once you would actually start using it. So that also means that the components can always be reused. So when you're done with one setup, you've done an experiment on there, you can move on to the other configuration and still use most of the components there. Okay, so that brings me to the last part of my talk. So in the next 5 minutes, I'll just explain briefly where we see Flamingo is going. And this is not just about us. So far, what I've shown you was really about how we can do sustainable microscope development in the lab. But now it's more about the sharing aspect of this. Because when the pandemic hit, we realized that these systems are actually portable. So I haven't mentioned this, but I just said that they are very compact. But I was able to take one of these systems home into my home office, and we learned that they can even be remotely controlled quite nicely. And so the idea was born to take these instruments, not just take a complex setup off the optical bench and have it in a more compact form on an optical bench, but rather, taking them with us wherever we go and to whatever collaborator we want to collaborate with. And so it was really this idea to also iteratively improve the system, right? So you come up with one design, you give it to the biologists, let them try it. If they don't like it, you take it back, you modify it, and that way, iteratively, you would come to a setup that happens to be sort of the best instrument for a given task. Right? And that's only possible if the instrument is portable. And that's very much in line with this concept that we proposed a few years back in Nature Methods, which is about this idea of putting the microscope into the hands of the biologists. So the idea, instead of building a fancy microscope in your optics lab and then inviting biologists to come with their precious samples to you to use the microscope, rather send them the microscope and that way give them access to the latest technology without, maybe sacrificing the biology or the precious samples if they have to travel somewhere. So this indeed works quite nicely. So the system is indeed portable. We can put it into these Pelican cases and ship them anywhere we want. We just came back on Friday from a workshop in Spain where we brought a Flamingo with us and did a live demo at the workshop there. So wherever you go, whether it's a workshop or a visit to your collaborator, you could potentially take such a microscope with you. So again, the idea is that you have an instrument, the biologist has an idea, and you communicate, and then you're able to ship this instrument to them. They get to test it, decide if they like it or not. And whenever they're done with the experiment, they would share it with the next lab in line. And so it's sort of like a library of microscopes that anybody could use and use for their needs, whether it's just testing it to find out if light sheet is the optimal technology for them or actually actively using it for a scientific project. And of course, it would only stay in the lab for a limited period of time before it continues its journey and travels somewhere else. So we've done this, for several different organisms at different institutions already quite successfully. We've been at the Marine Biology Lab in Woods Hole several times, and also in Madison. There are still a few flamingos that are being used by collaborators, like in Bill Bement's lab, for example. And so it's been it's been extremely successful. And of course, we are now expanding this to Europe, or at least to Germany for now. We also realized that most of the customization is really happening around the specimen. It's a lot about 3D printing the chamber, different sample chambers, sample holders, different embedding techniques, different incubation techniques, perfusion, whatever is needed to keep the sample happy. Most of the rest is typically always the same Flamingo setup. Okay, so one thing that I think is interesting to mention too, I mentioned this briefly, this idea of remotely controlling a microscope, because if you can remotely control one microscope, you can actually run multiple microscopes off the same computer. So essentially from my laptop here in my office, I can remotely control all the flamingos in the lab. And that also means that you can potentially remotely control a flamingo in my lab. And all these instruments stream the data to a central server. And so someone else, a collaborator of ours, for example, could analyze the data, or it could be automatically analyzed, and feedback could be then given to the microscopes. And that way they could be controlled by a smart machine, so to speak, so that the data is analyzed in real time, and then real-time decisions are being made as the data is analyzed. Good. So I'll quickly jump ahead and give you a summary here on sort of the last, the few flamingos that we've built so far. So there's 10 systems that were built in the US. We're currently building more here in Göttingen. Prices range between 50 and 150,000 euros, depending on how complicated they are and what kind of cameras and lasers they're using. And as I said, it's all about sort of the individual add-ons that are put on these systems, and they can be remotely controlled. So this is what it looks like in the lab. It's a lot of parts from the mechanical workshop and 3D printed parts. We also have a version in pink now, because the mechanical workshop was really excited about the idea of anodizing the parts in pink. They thought that would be a better fit for a flamingo. And this is now a new configuration, which is an inverted T system to look at like organoids in an FAP foil. And this is a system we are building for clear tissues, which is also a flamingo, just that it's built differently on an optical table because of the potentially toxic solutions in these chambers. So with this, I'd like to acknowledge people in my lab in Madison, in my former lab in Madison, especially people who were involved in the flamingo design and development, and then my current lab here at the University of Göttingen. And thank you very much for your attention.
Great talk. Thank you very much for that, Jan. So now, as is the tradition, we're going to go over to the quiz and see how people do with that. So Jan, if you wouldn't mind unsharing your screen, I'll switch over, and then we'll do Q&A afterwards. So here you should see the landing page for the Mentimeter quiz. The link was in the chat. Yeah, we've got a few people joined, got 5 people on it now. Looking forward to it. You get to win the wonderful fold scope for those of you who are quickest on the button. All right, okay, we've got quite a lot of competitors, so I think we should get going. And then after that, questions and whatnot. Okay, so here we go. We're going into question 1. Here are all our players. Okay, faster the better. Question 1. Light sheet microscopy is ideally suited to image single cells in a 3D culture, living embryos, cleared tissues, or all of the above. Correct answer, please. All of the above. Oh, gosh, we've got a lot of people here. So let's see. Let's see how we're doing after the first question. Sheety McSheety is in there for not the faster. Wolfmeister, Chamois, E.T., Ginger, Snuggles, Dragonite, and Scary. Scary is the winner with, I'm afraid, Lola and Kathy right at the end there. Right, so a bit quicker on the buttons, please, for the next question. Let's see if you can get on. Let's see if Sheety McSheety can do any better as well. All right, question 2. The goal in most light sheet microscopic experiments is to achieve what? To achieve the highest possible resolution, highest temporal resolution property, a 3D reconstruction of the specimen without damaging it, or uniform illumination of the entire specimen. What's the most important goal of those? And a 3D reconstruction of the specimen. Right, good. Most people got that, I think, without damaging it, in fact. Let's see how we're doing on the scores now. And Scary, and oh, Scary is very quick today. They all, and Dragonite, they're all still up there. Okay. Ginger, Snuggles, Spice World, and Wolf, you're snapping at Scary's heels. Let's see what happens next. Question number 3. Why does one do multi-view imaging? What's the reason for that? Is it because to achieve good resolution across the entire sample, to get a good reconstruction of the sample's front, to improve image acquisition speed, or all above? What's the most important reason for doing multi-view imaging? And the answer is, no, let's split the audience there. 8 people got the right answer. Let's see how the scores are doing now. That would be the
deciding question.
All right. Oh no, the wheel of death here. From premature end to our
we may have to announce the winner
at
a later point in time via Twitter or something.
Oh no, here we go.
Yay! There you go. Spice World. Look at that.
He's catching up.
Sorry, Scary.
Scary. Ginger and Spice World. Okay. I can see that they're taking time out from their work.
I think some people are working together here.
Is that you, Steffi?
No, I wasn't.
My internet connection. The Flamingo was designed to be what? A modular and portable microscope platform. Well, I think we got the message loud clear on this one. So, 13 answers on the modular and portable platform. Okay, let's see where we go with this. See if the spices are still up there. Spice World is still up there. Oh, Scary Spice is still ahead. Blimey. Is that an unbeatable lead? Let's see. Question 5. Just go for it this time. You might get you might win the Foldscope. You never know. All right. The name Flamingo comes from the color of the microscope parts, or is just a cool name for an innovative microscope, is an acronym, or it is used because it works best with fish embryos. And here we go. It's just a cool name for an innovative microscope. Right. Oh, we've got 5 people off the mark, so maybe things have changed. Let's see, big drum roll for the final winner here. And it's all scary is probably, yeah. Scary Spice, you are the winner. If you could just reveal yourself in the chat to the organisers, the fold scope will be on its way to you shortly. All right.
It's a bit of a letter downer to get a fold scope when you would really like a Flamingo scope.
Yeah. Let's go to questions. I'm just going to get the chat up here. Ah, James Manton is Scary Spice. Well, all right. Questions. We have a question from Valentin here. Do you want to unmute yourself and speak, or shall I just say it out for you? Let's see if Valentin is there. All right. Okay, I'll just read it out. Valentin, are there plans to implement other designs such as axially swept light sheet microscopy, recently published by Jörg Brewersdorf, or Reto Fiolka in a framework similar to the Flamingo project? I'll hand back to Jan.
Yeah, that's a good question, of course. So the idea with Flamingo is not that it stays the way it is, but is expanded by other techniques and new ideas. And you know, the axially swept light sheet is something that we're already doing for clear tissues, and it's just, yeah, it just takes time to implement it in the Flamingo in a compact and robust way, but it's certainly the way to go, at least for large specimens. So we're already doing that, but of course we're also inviting people to come up with more suggestions, whether they want us to implement them or whether they want to implement them themselves. We are happy to, you know, share the plans of the Flamingo or simply send them a Flamingo if they want to do some modifications to the system.
All right. Is anyone in the audience there want to stick their hand up and ask a question? Or put something in the chat. Let's see if anything else is coming up. I don't see any more, but actually, so I've got a question on the software side of things. Two ones there. I mean, how challenging was it to come up with a software package that would control all the hardware?
Yeah, unfortunately, there wasn't. Yeah, unfortunately, there wasn't any software available to us that we thought was good enough to run the Flamingo. So we actually wrote our own software. It's essentially two softwares. One is running on the Flamingo in a mini PC, simply to trigger all the components and run the experiment. And then there's an app that runs on your laptop with which you set up all the parameters, and then that sends a workflow file to the machine. So by the time you've started the experiment, you can then essentially close your laptop and walk away and have the system autonomously acquire the data. And this workflow file can also be written, by hand if you want to. So the GUI on your laptop isn't necessarily required to run the experiment. So yeah, this is all home written software that we designed specifically for the Flamingo. But it's the same software on all Flamingos.
Right, okay. So the second half of my question about software is image stitching. Do you have a sort of a go-to piece of software for that particular task?
We just use the software that's commonly available, like Fiji, open source software for most of the post-processing tasks, like the stitching or registration or so. So nothing special. Okay. It's just what most people are using in the Lightsheet community.
Right. All right. Any other questions from the audience?
What's the most exotic project someone has tried on your Lightsheet system?
I don't know. I think it's mostly been just some exotic samples from, I guess, mostly at the MBL in Woods Hole that people, go out on the beach and bring something back and say, can you image this? And, you know, only later we find out what species these actually were, but it's then all about embedding them and imaging them in the first place. And so it's those kind of places, I think, that are always exciting to go to with a portable microscope. So yeah.
Yeah, you're going to send one into space.
Question.
Yeah,
Sorry,
possibly.
Got a question from James Manton here. Hi, Jan. Thanks for a very interesting talk. For the combination of OPT and light sheet fluorescence microscopy, do you also do a fluorescence optical projection tomography acquisition to register the data sets?
Now, in this case, we would only use the OPT for transmission contrast, basically just label free. The fluorescence is typically acquired in the light sheet mode because it's, I think, superior to the OPT fluorescence. But the OPT is just something that can be acquired, so to speak, for free at any time throughout the fluorescence light sheet image pipeline. And the data sets, the OPT data set and the light sheet data set are registered because they are taken at the same time using the same camera. So there's no registration needed.
Okay, all right, thank you very much. And then I've got a question from Ben Sutcliffe. Hi, Jan, great talk. Following on from the software question, is the software available open source for others to use in order to control other custom systems?
Not so far. Maybe in the future. I think it's a very dedicated piece of software. It's not meant to be running, you know, it's not meant to be a universal microscope control software platform or anything. It's very dedicated for the individual hardware components that we chose. So it only works with that special kind of camera that we're using, etc. So of course, if people are interested in it, we are happy to share it. But the idea with, I should say, with the entire Flamingo project is not necessarily to hope that somebody else will be able to build another Flamingo. It's more about, you know, getting the funding to build, like that we built and with our collaborators build many Flamingos and share them with as many biologists as possible that may not be able to build their own microscope. So even if it was open source, most biology labs wouldn't be able to build such a system. So it's more about open access of such an instrument rather than sharing the technical details and hoping that many people will build them their own.
Okay, well, I guess unless anyone else has got any more questions, it's probably time for us to wrap up for the day. So with that, I've just got a thanks from MJG and all of the progress that has been made on light sheet in roughly 40 years. I've been aware of the general concept. Thanks for an amazing presentation. I think we all echo that thanks. So with that, thank you very much to our speaker for the day, and we'll see you all next week, we hope. All right, thank you very much.
Thank you all.
Oh, thank you.
Wonderful, thank you.
Excellent talk. Right.
Okay, I'll see you next week.
See you, Alex. Thanks very much. Thank you, Jess.
That was brilliant. Thanks, Jess.